Welcome to Season 5, Episode 27 of the Be Conscious podcast. Today we're going to talk about the coronavirus. Yes, the virus itself. Did you know that there are 6 known coronaviruses that naturally circulate in human beings? And all 6 have been known to cause only very mild respiratory disease, truly flying under the radar. Scientific studies have modeled the coronavirus species, its existence on Earth, that dates back long before we were ever inhabitants on the planet, long before human beings were here, and certainly long before we were ever counting time. These models of the corona species date back to as early as 8,000 BCE. So that's over 10,000 years ago, where ancestral lineages of the coronaviruses have dated back as far as 55 million years. Fast forward to the 21st century, to where we are right here, right now. Since 2003, there have been 3 zoonotic coronaviruses that have transferred to humans. So, a zoonotic virus is one that does not naturally occur or circulate in human beings, but usually has an animal species as a host. So, since 2003, there are now 3 of these zoonotic coronaviruses that have transferred into humans. The first was the SARS COVID, the original SARS, which is around 2003 timeframe. The MERS, you've heard of it, the Middle East Respiratory Syndrome, that was back in 2012. And now the current novel coronavirus pandemic that is caused by the SARS-CoV-2, like take 2 for SARS-COVID. Now, in each of these modern day pandemics, the animal reservoir remains unknown. There are suspicions, but it remains unknown. So let's take a closer look at the coronavirus itself. In this episode, we're going to dig a little deeper and look at what has changed in the modern era and who or what could be behind the pandemic. Here we go.
Welcome to the Be Conscious podcast, the show that brings you enlightened perspectives on the frequency of money, life's many challenges, and those heavy feelings that may be holding you back from living your best life. And now your host, Dr. Susan Nicholas.
We are in the 3rd wave of a novel zoonotic coronavirus saga, the Trilogy. According to the World Health Organization and the Centers for Disease Control, SARS, which stands for Severe Acute Respiratory Syndrome, was first identified in March of 2003 in southern China in the Guangdong province. SARS was first identified by the Italian physician Carlo Urbani, who he himself would succumb to the viral disease in 2003. There were a few atypical pneumonias reported in November of the prior year of 2002, but these and other subsequent infections were initially reported to the WHO because of a group of healthcare workers in southern China, a group of 22 healthcare workers, had this unusual pneumonia. This was then observed in Hong Kong, then Vietnam, and then finally reports into Singapore and Canada. So at the time, SARS was a new or novel coronavirus that had never before been seen in humans. By June of 2003, the virus was wholly contained. Basically, globally, there were no new infections, and the virus was fully contained within 6 months. There were no variants reported. 26 countries were affected, and it is said to have infected approximately 8,000 patients before it was fully contained. To this day, the animal reservoir for the original SARS infection is unknown. Now, I'm going to have the timeline and all of the links to all of this research in the show notes. Now, let's now look at the second novel zoonotic corona infection to affect human beings. That was MERS, for Middle East Respiratory Syndrome, first identified in 2012 in Saudi Arabia. Again, a novel coronavirus at the time. It was distinct from the SARS variant in 2003. Now, the MERS caused 178 laboratory confirmed cases and 76 deaths. So it affected fewer people, but it was more lethal. Primary cases were confirmed in Saudi Arabia, the UAE, Qatar, Jordan, Oman, and Kuwait. And there were some travel related cases noted in the UK, France, Germany, Italy, and Tunisia. Now, if we looked at the fatality ratio between the MERS and the SARS, it was much, much higher. MERS was 43% mortality, where the SARS had an 11% fatality ratio. So the timeline for the MERS was March of 2012 through December of 2013. And so within that time frame, which was nearly 2 years, just shy of 2 years, there were no new variants or waves of it, and it was wholly contained. And it caused 178 laboratory confirmed cases. Now, the MERS, the reservoir was searched for again, first in bats, and then they thought, well, maybe there was an intermediate species in the region. And so they looked at camels. Had camels spread this to human beings? Because when they examined the camels, they found what they called neutralizing antibodies to the coronavirus in a very high prevalence. But when they looked deeper at the time, these camels were also traded and um, moved from the Arabian Peninsula into Africa, and the African countries did not have that. They did not have the coronavirus, a species of it. And not every person that was in contact with these camels, even if they did have the neutralizing antibodies to corona, had not become infected. And so, again, there was no animal reservoir distinctly identified for the MERS either. So here we are in 2022. And we're in the third wave of this novel corona, this zoonotic coronavirus trilogy. And I dare say that this may go on and on and on, that I suspect we are not done with this. Because in looking at the modern era zoonotic novel coronaviruses, a pattern is emerging. And I think there's a link to this pattern that you may have heard in the news. And it has something to do with an enzyme called Furin, Furin. And this was originally identified in 1990. And there's a key, I believe, that unlocks this puzzle. And I believe that key lies in the creation of this enzyme called furin. Let's do a little investigation into what is this enzyme and how it could have led to the 3 zoonotic infections of the modern era. Stay with me.
Like what you're hearing so far? Make sure you never miss a show by clicking the subscribe button now. How would you describe your relationship with money? Did you ever imagine that your deepest feelings and beliefs about money could be preventing you from acquiring all the wealth that you desire? Click the link below in the show notes to learn more about Dr. Nicholas's highly sought-after Frequency of Money program to positively transform your relationship with money. This podcast is made possible by listeners like you. Thank you for your support. Now, back to the show.
SARS-CoV-2, the entry into human cells requires a sequential cleavage of that spike protein that we've heard so much about, that's on the envelope, the outside envelope of the coronavirus. The spike protein is a glycoprotein. And there are areas of this that have been named, of this protein that have been named. They're called the S1 and S2 areas, and an S2 prime cleavage site that mediates the membrane fusion of that spike protein to human cells. So, if you listen to my prior podcast or if you've learned about the spike protein, you recognize that these zoonotic coronaviruses, these novel coronaviruses, on their own and in nature, for over 55 million years, they have not been able to infect human cells. But all of a sudden, they have this capacity. And I believe it's linked again to this enzyme called furin. And we'll talk a little bit about that. So basically, there are two sequential steps that are required for that COVID virus to actually enter human cells. It won't do it on its own. It's not designed to, not in its natural state. It has to be manipulated or modified, if you will. The virus has to be modified to be able to enter human cells. Now, that spike glycoprotein, that S1, S2 site, has to be cleaved. It has to be broken to activate it. To begin that two-step process for that spike protein to attach to human cells and to literally dump its genetic material inside of the cell and overrun the normal cell function. This S1, S2 site is cleaved by an endoprotease. It's a protein enzyme called furin. Basically, furin is an enzyme protease that is utilized in humans and other animals to activate proteins. So basically, furin is used to activate that spike protein. That spike protein alone cannot enter human cells. It has to be activated in a two-step sequential process. And what mediates that reaction is a protein enzyme called furin. You see, many proteins, and this is true in nature for all mammals and other types of living beings, some proteins are inactive when they're originally synthesized. And it requires an enzyme process to really catalyze that reaction. And with the novel coronaviruses, these zoonotic coronaviruses that have never before in the history of the world affected human beings, are activated by furin. So what it does, it in that cleavage process, it actually removes a section of that genetic code in order for that to become active. So basically, furin will cleave that S1, S2 section of the spike glycoprotein and activates those proteins. So when that S1, S2 site is cleaved or deleted, the virus has a selective advantage to enter and infect human airway cells, human airway epithelial cells. It attaches to something called the ACE2 receptor. These are in the lungs. So the SARS-CoV-2 or the coronavirus enters human cells by their spike glycoproteins that must be cleaved to mediate cell membrane fusion. And like the other novel coronaviruses, the zoonotic reservoir of their SARS-CoV-2 is yet to be determined. Now, you may have heard in the news cycle that there's a question of bats being the reservoir. But what we learned from the MERS infection is that humans and bats have very little contact, and certainly not globally, very little contact. And so there must be an intermediary, they thought, and that's how they looked into the camels and still came up with inconclusive animal reservoir. So this leads us to something that you may have heard called gain of function research. This topic has been politicized because it is gain of function research in the virology lab in Wuhan, China, that has been credited, if you will, for leaking the coronavirus into the human population. So in comes gain of function research. So if we look at the timeline of the ability to manipulate viruses, we have the creation of Furin, this enzyme protease, in 1990. Then we see about a decade later, we see the first SARS. Then about 9 years after that, we see the MERS. And then 8, 9 years after that, we see SARS-CoV-2. What I suspect is happening is that, for better or worse, the research of manipulating viruses has gotten better. So basically, we've gotten better at changing the genetic code of viruses to make them more transmissible into human beings. So what is gain of function? Gain of function research, it refers to the serial passage of microorganisms to increase their transmissibility, their virulence, their immunogenicity, and the host tropism by applying selective pressures in culture. So when you apply a selective pressure in culture, that means it's done in a laboratory. This is not done in nature. So the rationale behind this gain of function research is that it allows for disease control measures to be planned. You've heard of the pandemic. It allows for disease control measures to be planned, and it allows for potential vaccines and therapies to be created and explored. This gain of function research utilizes something called CRISPR, CRISPR, which is a gene editing technology. And it essentially gives proteins like that spike glycoprotein a new set of instructions. The gain of function research refers to the virus being taken from its animal reservoir, its animal host. It is then genetically altered in the laboratory to make it more transmissible in human beings. So before we go on into this gain of function research and why it is so controversial, I mean, just by learning about what it is raises red flags, I think, for most of us. But let's talk a little bit about what is host tropism. Because host tropism is what keeps a virus species in a certain host. So it's what keeps the bat coronaviruses in bats. It's what keeps dog coronaviruses in dogs, keeps human coronaviruses in humans. Just for example. Because viruses don't typically have the instructions or the genetic code to trans-infect species. And for good reason, right? As human beings, we are not bats. We are not dogs. We are not birds. And so our immune system is not set up for viruses that typically have zoonotic or animal hosts. Host tropism is the infection specifically of certain pathogens, like the coronavirus, to particular hosts, like bats, and specific tissues in that host. Now, in this whole coronavirus trilogy, from the first SARS in 2003 to the MERS in 2012 to the coronavirus in 2020, humans are now the new host. And that tissue has been the lung epithelium. And now through this saga of COVID-19, we are seeing other tissues that are being affected, like the cardiac tissue. And so how is this possible? So if we think of the timeline, in 55 million years, there was no change in the zoonotic coronaviruses. They were never able to infect humans. In a very short time, they've come into human populations in each of the novel coronavirus pandemics. The animal reservoir has been unknown. Miraculously, they come into human people and it devastates populations. What's very interesting about viral mutation is that the mutations a virus acquires are deleterious to the viral function. It usually decreases their function. However, it has been recognized that some mutations, particularly gain of function mutations, can enhance the function. So this is very, very important. The FDA requires that animal testing be performed for vaccines before human trials can go ahead. However, as viral tropism toward the model species is unlikely to exist already, so because of viral tropism, it is unlikely that an animal species will affect humans. In cases where human viruses are under investigations, those strains have to be created, they have to be generated. Those viruses have to be modified in a laboratory for them to infect a human. Those animal species, those animal viral species have to be modified. They have to be created. And this can be achieved using gain-of-function research, where the virus is passaged through animals, allowing molecular determinants of transmissibility to be identified, and it allows vaccines under investigation to be tested. And again, I'll have the links in the show notes. So it is known that this gain-of-function research, what it does is figures out how to get animal species of viruses into human beings. How does it get them to infect human beings so they can develop vaccines and therapies and plan pandemics? Doesn't that sound strange? Like you're creating a problem that doesn't exist? So this research, there's ties to NIH funding. And one of the reasons that this is under attack is because of the dangers of this type of research and ties to money from the United States. And so I don't want to make this a political argument, even though I know it's highly politicized. But with 5.5 million deaths worldwide attributed to COVID, either you're dying of COVID or dying with COVID, notice the narrative. Listen carefully. Science is very complex and it's incomplete. And the narrative relies on the general public's perceived lack of understanding of science. And I believe this is to hide the truth. Now, you may not be a scientist, or perhaps you don't understand all the terminology. That's okay. But what you do know is when something doesn't feel right. In COVID-19 alone, we have seen several variants of it affecting different tissues and having varied disease profiles, varying transmissibility, varying virulence. How is this possible in a single 18-month period in a single coronavirus species, something that can change so dramatically in a very short time that was stable for more than 55 million years? What is happening now? And it is technology. It is gain-of-function research, and it is leaking of these very harmful viruses from a laboratory setting, a manipulated laboratory setting, into the human population, whether intentional or not. But this is viral gene manipulation using what we call gain-of-function research. I think it's really important to follow the money. The NIH has been implicated in funding gain-of-function research in Wuhan, China. You may have heard that the NIH says, hey, we didn't directly fund this research. We had a grantee that funded the research, and that grantee of those NIH research dollars was EcoHealth. The EcoHealth Alliance, a US-based NGO, non-governmental organization. And EcoHealth is blamed for funneling money, US funds, to Wuhan, China. So in the 2014 to 2019 timeframe, the NIH granted the EcoHealth Alliance $3.1 million, which directed $599,000, just a little over $500,000, to the Wuhan Institute of Virology for bat coronavirus research. This is where this implication is. So whether the NIH, the US government, was knowingly or unknowingly involved in this research. That's what's going on with the money. So, just for context, the coronavirus circulates among mammalian and avian reservoirs, including human beings. But those zoonotic coronaviruses that we're now seeing in humans live in bats, pigs, dogs, rodents, and birds. They do not live in human beings. They are genetically altered to do so. If you've heard the word zoonotic spread, that means it occurs between animals and human beings. And when we think about increased transmissibility, that really refers to how it is spread. So typically, the coronaviruses are spread from droplets. It's from someone coughing or sneezing or being very close contact. You've heard of bloodborne illnesses, waterborne illnesses. This is in the droplet or the waterborne arena, where it requires you to be in relatively close contact. You've heard of nosocomial infections. People get them in hospitals, being on ventilators. It's the idea that you're getting an acquired infection because of, let's say, contaminated instruments, but very, very close contact. It's not by walking down the street or sitting next to someone or being in school or not wearing a mask, for instance, unless someone actively coughs or sneezes or sprays droplets on you. So how do you increase transmissibility of a virus like that? You have to genetically alter the virus to spread through the air, to be airborne. And so when we look at these new waves of the SARS-CoV-19, we have to think about how it's being transmitted and how can that change so quickly when evolutionary biology, evolutionary virology, it does not occur in nature. When you hear information like this, when you hear the news cycle and you hear the narrative, how does it make you feel? Do you feel informed? Do you feel controlled? Do you feel empowered with the information? Ask yourself, how does it make you feel? Does it leave you curious to learn more, or does it leave you afraid? Does it add to the confusion or leave you with more clarity? These episodes are intended to leave you informed, curious, questioning, and empowered. And please, if you have questions for me, something resonates with you or doesn't, I'm happy to take comments and questions at info@susannicholas.org. And remember, whether you are coming or going, looking inside or out, remember to be conscious.
Thanks for listening to the Be Conscious podcast with author, TEDx international speaker, and conscious coach, Dr. Susan Nicholas. Be sure to connect with her to stay up to date with her latest news and works. Learn more about her conscious speaking platform at susannicholas.org/speaking. Stay tuned for the next thought-provoking episode. And whether you are coming or going, looking inside or out, remember to be conscious.